Hello, Mary. Can you hear me? I can hear you talking.
Hi.
Hi, how are you doing?
Good, how are you? My computer was muted.
Okay, gotcha. How was your weekend?
It was really good. How was yours?
I was finishing up some home renovations and I was breathing in, I guess, too much sawdust and I sneezed a lot,
Oh,
which when you have bruised ribs, that is not an optimal situation.
yeah, that sounds pretty painful.
Yeah, it's not very much fun. I'm walking along feeling fine and then I'm on the ground in pain. And my partner's like, "What's wrong?" I'm like, "Just sneezed, just sneezed." So anyway, yeah, I'm still. I was making pretty good progress. I was feeling pretty good on Thursday, and then on on Saturday I started sneezing, and it's just really unpleasant.
Yeah.
So we'll see. Anyway, I was rehabilitating a door, and now that door is finished, and it'll get installed tonight, hopefully, and then I'll stop sneezing.
That's
So
good.
anyway, who else do we have? Winston, Branson. Jackson.
Yep.
So what is the shack about?
So I'm only made it a couple chapters in, but it's pretty much um, about this family, and they went on a trip, and like one of the main character, his daughter got abducted from the campsite, and so now they're like working to try and find her, and that's pretty much where I've made it so far.
All right.
But yeah.
All right. It's technically 9 o'clock, so I'll go ahead and get started. Just a couple of kind of housekeeping issues. Um, those of you who have submitted some food blogs, thanks for that. It seems like some of you have been having some difficulties seeing the results. My advice would maybe be to log out of WordPress and then log back in, and that might help. Did somebody have a comment? No, I thought I heard something. So that's actually sometimes necessary to do, unfortunately. I've read a couple of them, and I didn't this weekend get around to putting my first post in yet, but I'll hopefully get that in later today or maybe tomorrow. Mondays are fairly busy for me because we have research meeting in the biology department in the afternoon, and so we'll see how diligent I am at getting that done. Second thing is be checking the Moodle site on a regular basis. There's a way to actually turn on notifications from Moodle, and you might or might not want to do that, depending, because you might just get tired of getting notifications from Moodle, which I get. But for example, let me share my screen with you for just a minute. This is today's PowerPoint that I was looking at before class, and that's R. I was also doing some work in R before class. let's go to Moodle. No, not Google, Moodle. So if you go to Moodle, you'll see that a couple of things have been added since last Friday. If you click on lectures and screencasts, this takes you to a little post where basically, oh, it doesn't take you to a post, it actually takes you to the YouTube site. So here you can find all of the recordings of the Zoom meetings that we are having. And so they'll be posted later on the same day that we have lecture, generally speaking. And so all you have to do to access those is to click on that thing. It's just a web link built into Moodle. And then also I've added materials for today. So you'll see that there's a basic plant structure PowerPoint on there for today. If you have notifications activated on Moodle, you will get a notification when stuff gets added so that you can go and get those things as you wish. So I think that was all I had in terms of kind of housekeeping things. Do you guys have any questions?
I have a question.
Okay, go ahead.
Yeah, who's that?
When do you want like the food blogs to be posted by each week?
Okay, so
Us too. There's like there's like a time frame is like I don't know, August something to like September something. Can they be posted like between there?
yes, so that's the 2 weeks that span the time for that to be done. Let me pull up the syllabus because the answer actually resides in the syllabus. So let me go and get the syllabus. If you scroll down into the course requirements section, after all the amazingly long course policies, there's a response to food journal prompts. So you need to contribute to a course blogging site. You will be given a series of prompts to respond to and need to post original content to the blog post at least twice per week, unless otherwise noted. I told you in this assignment that I only need you to make one post this week, but I also want you to respond to 2 other posts while you're on there. You cannot leave the assignment until the end of the 2-week period and then post 4 original posts and 4 comments the night before the due date. The reason is that, for one thing, it doesn't give anybody else anything to respond to. If everybody goes on the night before the assignment is due, post 4 blog posts and 4 comments all at once, because you'll be able to post your 4 original posts, but there won't be anybody else's posts to comment on. So the idea is to have this be a running conversation over the 2 weeks of the blogging. So I think right now there's something like 5 posts on the food blog. And so when you go to add your post, you can add your post, but then while you're there, you can read a couple of other people's posts and post responses to their posts. I, for example, have never been a fan of mashed potatoes, especially the way my mother cooked them when I was a child. So I should put that post on. Who was the mashed potatoes post?
That was me.
Okay, yeah. So you are certainly more fond of mashed potatoes than I am, but I should share that comment with you on the blog rather than here in class. So there is no do it by this date, but sprinkle them throughout the 2 weeks for 2 reasons. It takes the pressure off of you, and for a second reason, it also seems more like an ongoing conversation rather than just a course assignment. Does that answer the question adequately? I realize you're like, what's the date? And I just said, well, there isn't a date, just don't do X. So, but hopefully the spirit of the thing is intact. So feel free to ask those questions, but also I've tried to foresee as many of those questions as possible in the syllabus. So you can almost always probably find the answer to something like that in the syllabus as well. But it's there. Other questions? So the other thing that I want you to be paying reasonably careful attention to is the schedule. So Oh, I guess it was already open. It is already open on my computer. So for example, today we're scheduled to talk about plant biology. We need to finish up some thinking like scientist stuff from the previous session. Then we'll be talking about plant biology. In order for us to talk about plant biology effectively, you need to have delved into chapter 3 of the Plants and Society book, because that's the material that we'll be talking about today. And I had also asked you for Friday to have read the introduction in chapter 1 of Michael Pollan's Omnivore's Dilemma. So we're going to use Pollan's chapter 1 to kind of set the frame for why we're going to be doing essentially 3 weeks of plant biology. And the reason that we're going to be doing this is because in chapter 1 of Pollan, which we'll get to here in a little while, he basically covers so much plant biology in the first chapter of the book that it's going to take us all the way until exam 1 to give you the knowledge base with which to evaluate the things that he's talking about in chapter 1. So that's the reason that you have a Pollan chapter here at the beginning, and then we don't revisit Pollan again until later in the semester. We're going to spend what seems like an insane amount of time on chapter 1. So be paying attention to the schedule. It's on the Moodle site, and let me know if you're having trouble accessing it. One person had some trouble accessing it. So, other questions?
Where can you find that schedule?
Oh, so on the Moodle site, it's in the first day of classes stuff. So right here it says syllabus and schedule. So you click on and it's right there.
Awesome.
And over the semester, I'll be adding more readings in the reading section as we get to those portions. And when I do that, I'll update this and I'll put a new date on it so that you'll know that it's an updated file.
Thank you.
Sure. All right, other questions? All right then, we were talking about thinking like scientists, and I gave you a task over the weekend. What was that task? I don't really want it to be in pre-interview. Does anybody remember what the task was? Oh, crud. I think I may have lost my audio. Jackson, are you speaking?
No, I'm not.
Okay. All right. Okay. So I can hear you. Okay, good. So let me try and share screen again. All right. So what was the task that I gave you guys over the weekend? We were looking at this figure. We were seeing that coal production is going up at the same time that coal employment is going down. And I asked you to ponder something.
Weren't we supposed to think about like graphs and how we extract coal in relation to coal miner employment?
Yes. So one of you mentioned towards the end, I forget who it was, one of you mentioned that probably something technological changed that allowed you to gather more coal with fewer and fewer coal miners. And I asked you to think about that and do some research over the weekend about that. Did anybody do that? Olivia, did you just nod your head?
Yeah, I read about how we extract coal from the ground, just the technology that goes behind it.
Okay, talk to us about that. What did you learn?
I just learned about the mining process and how the equipment that they use to drill coal instead of having men inside the caves personally drilling all the time.
Okay, so back in the old days, we would dig shafts into mountains, and coal miners would go down into these tunnels in the ground, and they would extract coal and haul it out on little railroad tracks. And it was a very dangerous situation because of mine collapses, and also it's a great way to get black lung. And back during the 1970s, coal mining technology changed. Did you see anything about how it changed?
A little bit how really the men don't need to be inside the mines anymore because of the equipment they're using.
And so what methods do we use now?
I believe drilling, a type of drilling.
It's no longer drilling. Anybody else find anything?
I've seen videos, but I honestly have no clue what they call it. But they use a chain, and they like it's like two men, and each of them snap on the chain, and it tightens it. And I don't know what it's called, but it's pretty cool though.
No, I haven't seen that. The big thing that changed, and this started in the 1970s and it picked up in the 80s and 90s, is a thing called mountaintop removal. And so a lot of these coal mines were located in the Appalachian region. And if you go and if you search for mountaintop removal, you basically see that the way we do it now, here's a good visual of it. We basically dynamite the top of a mountaintop off until we reveal the coal seam. And then one guy with a crane with a gigantic scoop on it basically scoops up the coal seam and dumps it into these gigantic dump trucks. And so here's another example of mountaintop removal. Here you can see all of the tilings from the mine that are going into the valley between this mountain and the adjacent mountain. And so they went from sending miners down into tunnels to basically just removing the mountaintop that is on top of the coal seam and then scooping it out with large machinery. And so all of a sudden, because you can have these gigantic dump trucks and these gigantic scoops, you can have a mine that used to be worked by hundreds or thousands of miners that can be worked by a relatively few number of people. And so it allowed mining companies to really improve their efficiency in terms of how many miners do you have to get a certain amount of coal out of the ground. And this has become more and more prevalent throughout Appalachia, which explains the shape of this particular curve. And so when coal miners are upset about losing their jobs, the only people that they have to blame for the most part is the mining company executives who have basically shifted to more labor efficient ways of getting coal out of the ground. There are all sorts of environmental reasons why you might not want to do this, like just tons of environmental reasons why you wouldn't want to do mining this way, but it ends up being much safer for the miners, but it also requires many fewer miners to do the job, to do the work. So the point of all of this isn't to harp on mining. The point of this is to think about kind of how a scientist might think about other issues, not science issues. And so the way the scientific method works basically is kind of rigidly stepwise. It begins with the process of observation if you're doing kind of pure science, which we've just kind of skipped over the observation part in these examples. And once you have an observation, you generally generate some sort of question. And this question has a set of possible answers, and these answers are hypotheses. So when I think about hypotheses in science, a hypothesis is just a proposed answer to a question. And in that PowerPoint, we were basically starting with a hypothesis. Let me go and get that figure up again. So the first hypothesis that we had was stated as a question. This question, do tariffs on China affect the price of farm commodities? Well, the answer is that tariffs do, or the other answer is tariffs don't, and something else affects the price of farm commodities. And so a hypothesis is just a proposed answer to the question. Then what we do is we go and find data that are relevant to the question and the hypothesis. In some cases, scientists do things like experiments. But if you are a social scientist or an economist, you could think of yourself, if you're an economist, as being a form of scientist. You can also get observational data. Both of these things work equally well in the context of kind of what we're doing. And then we look and see if those data are consistent with the hypothesis or not. So we analyze the data in light of what the hypothesis is, and then we reach some conclusion. And in the case of tariffs, we found that tariffs don't have nearly as big of an effect on farm prices as other things do. And in the case of coal employment, changes in technology have way more to do with coal employment than probably anything else. And so we can evaluate each of these ideas by basically finding the relevant data and then asking, well, if this hypothesis is correct, what would those data look like? And then looking at the data, and if the data look like what the hypothesis would predict, then you would say there's some validity to that hypothesis. But if you look at the data and find that they don't match up with the predictions of the hypothesis, then something else must be driving those data. And then you can start erecting alternative hypotheses that might account for those data. What you can't do is just say something and then not care whether or not it actually conforms to reality. I mean, I guess you can do that. It's just that that's not my preferred way of going about things. And that's not scientists' preferred way of going about things. And so in this class, when I say something or when you say something, I want us to always be able to go to some external source and be able to say, okay, that thing that we just said conforms to reality or doesn't conform to reality. Because if we don't have some common basis of fact to begin with, there's really no way of talking about the things that we're talking about with any sort of intelligence. Questions about that? Can you guys see the board adequately?
No.
No. Do I need to move closer?
Yes.
What was working for me is that I just go up to your screen and then click the three dots and click pin video, and then it makes the video bigger, like your screen. So then I could be able to read the whiteboard.
Oh, okay.
Yeah.
All right.
That works for me.
Okay. Well, I can also move closer. So just give me a sec. I'm going to unclick my computer and I'll just move closer. Is that better?
Yeah.
Okay. All right. So let me close this thing down. Go to your participants tab on Zoom and click it, yes, if you've read the chapters in Pollen, the introduction and chapter 1, just so I can see what I'm working with. Okay, if you haven't read it yet, by all means, do read it. So these questions will be for people who have read it. In the introduction of Pollen's book, he makes the claim that we have a national eating disorder. What's his basis for saying that? I have headphones on so I can still hear you when I walk away from the computer.
I would say that I think he says that because we don't really have like a stable eating thing, and like the media will kind of control what we eat, so we'll just bounce around from different types of diets. So that's kind of what I got out of it.
Okay, so one of his claims is that we lack a stable food culture. And what's the example that he uses to illustrate this?
It was that meat diet, or
Oh yeah, the demonization of bread during the 90s when the Atkins diet came out. This idea that bread was going to kill you and lead you down a path of destruction. As he's making this case, he talks about the American paradox. Do you recall what the American paradox is? He contrasts it with the French paradox. Okay. Anybody else remember what the American paradox is?
Wasn't it where he was talking about how like we're always trying to eat healthier and less, and we're always getting like bigger, but like the French are eating like really unhealthy things all the time, and they're known to be pretty skinny?
Yeah, and so we in America refer to the French paradox, which is basically people sitting around at street cafes drinking wine and eating cheese, and they're as trim as can be, and we refer to that as the French paradox. But he calls attention to the fact that they probably think of America as the American paradox, which is a bunch of people obsessed with eating healthily who are among the most unhealthy people on the planet. If you look at data on rates of obesity and things like this. And so we think of the French paradox, well, they could think of the American paradox. They're both kind of equally valid. So he begins with this idea that there's something wrong with American food culture. And so he goes on this exploration of food culture. And in the beginning of this, he gets at this idea that one of the problems that humans have in terms of their relationship to food is that human beings are omnivores. And so the title of the book is The Omnivore's Dilemma. And I'm just going to warn you, the first question on every test you take in this class, the first question on the test will be, what is the omnivore's dilemma? So today, who can tell me what the omnivore's dilemma is?
The author says that basically it's just a long answer to what we should have for dinner. I mean, unless you're just
but why is it a dilemma?
just because we don't know, or I mean, we have trouble with knowing what to eat.
Why do we have trouble knowing what to eat? Somebody else. Mackenzie, maybe.
Sorry, I'm trying to figure out Zoom still. I mean, I'm really bad at quoting things off memory, but like a lot of issues, like it talks about fast food as well. So it's like you can eat, like being an omnivore, you can try to eat healthy, but it's not always gonna be like beneficial to you, maybe type sense kind of thing. I don't know. I'm really bad at quoting things.
That's okay. Let me see where I can find it in the book. I have to go back a little bit. I was reviewing things before class. Ah, there's the koala. I just passed the koala. So in 1976, there was this paper by Paul Rosen, and in it, it looks at rats foraging. And he talks about here the koala. If you think about a koala, The koala doesn't have to worry about what to eat. If it looks and smells and tastes like a eucalyptus leaf, it must be dinner, because a koala is an example of what we refer to in biology as a dietary specialist. An omnivore, on the other hand, what does the word omnivore mean?
Eats meats and greens, like plants and meat based, pretty much.
Yeah, more broadly, you can eat anything. You can
Yeah.
eat a burger, you can eat a corn on the cob, you can eat a Twinkie, whatever a Twinkie is made out of. And so the challenge of an omnivore isn't not being able to eat things. This is the situation with the koala. The koala only eats eucalyptus and would probably get sick if it ate anything other than eucalyptus. And so the question is, the omnivore's dilemma is basically this. How do you decide what to eat if you can eat anything? And this is the dilemma. We can eat and digest anything. So then the question is, what should we be eating? Because we can eat anything. Should we eat a nice healthy meal of, skinless chicken breast with a side of broccoli and edamame, or should we be eating a Snickers bar and a key lime pie slice? Well, we can eat both of those. Both of those have calories. One of those diets is more healthy and good for us than the other of the diet. But the fact of the matter is we're capable of eating both of those things. And so that's the dilemma that we have. And it's the same dilemma that rats have. How do rats decide what to eat? Because rats also are omnivores. They can eat a wide variety of different substances. And so when we think about the omnivore's dilemma, this is the dilemma. What should we eat when we can eat anything? So those are the two big points of the introduction. And so when we get into chapter 1, though, things start to really accelerate fast. Who can tell me what some of the major themes are in chapter 1? What are some of the major ideas that Paulin talks about in chapter 1?
I think the biggest takeaway I found was that corn is literally in everything. Like he talked about how it can be in foods. It's in it's what we feed the animals that become our food. It's in literally everything.
Okay, you're going to find that the whole first half of this book is focused on corn. And the reason is that corn is in everything. Corn is in all of our food. Corn is what we feed our meat animals that we then eat for meat. Corn is actually in the adhesive that holds the paper on drywall in your houses. Corn is in the ceramics that wraps around the metal parts of the spark plugs in your car. Corn is in your soda bottles, in your plastic bottles that your pop comes in. Corn is in the pop that is in the bottle that is made from corn, et cetera, et cetera. So certainly one big theme that is in this book at the beginning, in the first half of the book especially, is that corn is ubiquitous. It's everywhere around us. It's in everything we eat. It's in all of the consumer products that we buy. Another idea that is in this first chapter.
Maybe that everything is connected in a food chain, he says.
Okay, so there are connections among things in food chains, although that becomes more of a theme later in the book, but certainly. What else? I'm thinking around page 22 is when he starts drilling down into corn more so. Can you guys see my book? When he talks about corn walking, what is he talking about? Is I think one of the most difficult parts of the omnivore's dilemma is actually section 2 of chapter 1.
Is he talking about the correlation with carbon?
Yeah, who's that? Marcus?
Yeah.
Explain that to us a little bit.
Like it says, one would expect to find a comparatively high proportion of carbon, depending on how much corn you're taking in.
Yeah, but it's a particular kind of carbon, isn't it?
C4.
It's not C4, it's carbon 13 versus carbon 14.
14, yeah.
I'm sorry, carbon 13 and 14 versus carbon 12. So by page 22, he has gotten into an area of ecology known as stable isotope ecology. You guys remember what an isotope is? I'll take it from your silence, that's a no. Okay, so an isotope is, so when you think about carbon, carbon has 6 protons, 6 neutrons, and 6 electrons. Protons and neutrons are heavy, and electrons weigh hardly anything. And so when we think about the mass of an atom, we don't even really think about electrons. But but the protons and neutrons weigh about the same. And so if you have, and they weigh about 1 mass unit, and what's known in chemistry and physics as a mass unit. And so carbon, which has 6 protons and 6 neutrons, has an atomic mass of 12. And so if you were to look at carbon on a periodic table of the elements, it would give you the mass of carbon. But what you would find is that the atomic mass of carbon is 12.011. It's not 12. And the reason for this is that most carbon that we see in the world, can you see this carbon up on the board behind me? Most carbon has 6 protons and 6 neutrons. And if all carbon had 6 protons and 6 neutrons, then the mass would be 12. But some carbon has 7 neutrons, and some carbon has 8 neutrons. And this is where we get carbon 12, carbon 13, and carbon 14. It's because different isotopes differ in the number of neutrons that they possess. And when they do that, they weigh different masses because of the additional mass of the neutron. So we don't just have carbon 12 floating around, we also have carbon 13 and carbon 14. So in order to understand this little section of the chapter, you have to know something about isotopes from a chemistry class that you took someplace else. But the other weird thing is that C4 plants tend to capture out of the atmosphere more carbon 13 and carbon 14 than C3 plants do. And they do that because their photosynthetic system is different than the C3 plants. And it's different in a way that captures more of the total carbon. And as a result of that, it captures more of the rarer isotopes of carbon. So all of a sudden, to understand this one section, you have to not only understand a little bit of chemistry, but you also have to know a certain amount about photosynthesis for you to truly understand the complexities that he's trying to get across to you in this chapter. And this reveals one of the challenges of writing a book like this for the general public, is that you can't expect the general public to be following you completely as you're making these arguments. So I'm going to erase what's on the board right now and put up the main sections of chapter 1. So in chapter 1, by page 22, we're talking about stable isotopes. and the photosynthetic differences between C3 and C4 plants. By page 24, we're talking about the evolutionary origins of corn. Corn came from a plant called teosinte in Mexico, and it looks very different from teosinte. And one of the reasons that it looks different from teosinte is given to you on page 27, where it talks about the genetic modifications that occurred that converted teosinte into corn. And in the process of talking about that, by page 28, there's a coverage of mating systems in plants, essentially how plants have sex. And so there's information about plant reproductive biology. And at the very end of the chapter, you get to page 30, and he's shifted to the genetics that are involved in hybridizing corn as it's hybridized in the United States in the 21st century. This is just chapter 1. So, one of the challenges of reading chapter 1 of The Omnivore's Dilemma, a book written purportedly for the common reading public, is that there's just a ton of science in just chapter 1. And so if you go and look at the schedule, let's pull up the schedule again. You can see that here at the beginning, we're going to be spending time in plant biology. We're going to talk about genetics. And so the plant biology that we're doing is we're going to do some anatomy. Then we're going to talk about energy capture and basic photosynthesis. Then we're going to talk about C3 and C4 photosynthesis. So by the end of this week, this second part of the chapter dealing with corn walking should be abundantly clear why he's making those kinds of points to you. Then we're going to talk about mitosis and meiosis, which is necessary to understand genetics. And then we'll be working up to understanding plant and animal breeding, which then helps us understand the other part of chapter 1. And so essentially, the first 3.5 weeks of this class are all focused on trying to understand the intricacies of chapter 1 of The Omnivore's Dilemma. So when you ask yourself, well, why are we learning all this stuff about plants? The reason we're learning all this stuff about plants is so that we can understand chapter 1 of The Omnivore's Dilemma. So if you felt that chapter 1 was a little bit of a hard read, it is because he has packed in a bunch of stuff into chapter 1. And our goal for the next couple of weeks is to make that stuff clear to you. Questions about that? All right. So let's begin. Let's see. You guys can still see my screen?
Yes. Hey, actually, I do have a question.
Yeah, Marcus, what's up?
Are you giving points if we read or if we talk during class? I don't know.
No, there is no participation. I am keeping track of who's talking. I have a little grid here, simply for my own information, and also so that when I see that Mary and Blake have asked a lot of questions, I can go down and say, hey, Mackenzie, you said that you read the book also, but you haven't said anything. So in the same way that I would be kind of monitoring informally who's talking and who's not during any given class session, if we were in person, it's a little harder to do that online. So I'm kind of keeping track of it on a sheet of paper, but there is no participation grade in here. I just expect you to participate as you are doing.
Okay. All right. Thank you.
So of course, in order to participate, we have to come prepared to participate, and that's the main thing. So if you haven't read chapter 1 yet, or chapter 3 of the Plants and Society book, everybody should do that. All right. Okay, basic plant biology. All right, can everybody see this? I can't see you at all because I've reduced the thing, so I can't see heads or anything.
Yeah.
Okay, all right. So what we're going to be doing here is we need to understand some basic biology of plants. And so we're going to talk here at the beginning about basic structure of plants that help us distinguish between monocots and dicots. Monocots are the group of plants that corn belong to, and dicots are the group of plants that soybeans belong to. And we're going to talk a lot here at the beginning about corn and soybeans because those are two dominant crops in the American food supply chain. And so understanding the differences between monocots and dicots is going to be useful to us. In the process of doing this, we're going to talk about leaf structures that have to do particularly with fluid transport in plants, because those leaf structures that have to do with fluid transport also have a lot to do with photosynthesis. And so our goal in going through this structure, the knowledge of the structure of plant bodies, is so that we can then have a better understanding of photosynthesis, because understanding photosynthesis gives us this key to why corn is different from soybeans, why corn is grown in some parts of the country and not in other parts of the country, and the same is true of soybeans, and why corn harvests different kinds of carbon than soybeans do, for example. And so we will be looking at these plant organs as they relate to fluid transport, nutrient absorption, and our big focus is going to be to focus on photosynthesis. So as we go through the next day or two, really 3 days, it's all focused on these things. What are plants built like? What does that have to do with photosynthesis, plants getting the nutrients that they need, and getting the water that they need? So this is what we should be thinking about as we go through this process. So plants are like any other organism. They're made up of a bunch of cells, but inside of cells, there's a lot going on. And plant cells are in some ways fundamentally different from animal cells. And so one of the ways in which they're different is, can you see my arrow on my screen?
Yes.
Okay, I can also give myself a laser if that would be useful to you. So here we have, so this thing in the middle of the plant cell called the central vacuole. This is something that is very characteristic of plant cells. It's just a large fluid-filled sac of a membranous sac inside of the plant cell. And then another way in which plant cells are different from animal cells is that there is a cell wall made up of cellulose. Cellulose is just a bunch of glucose molecules bonded together in a particular way, but it forms a semi-rigid wall that goes around the cell. And the cool thing about this is that because the plant cell has a bunch of materials inside of it, water will tend to want to move from outside the plant cell to inside the plant cell. As it does that, it fills up this central vacuole with fluid, and that fluid then pushes out against the cell wall. And when it does that, it's kind of like inflating a bicycle tire. When a bicycle tire has very little pressure inside of it, it's flaccid, and you're rolling around on under pressurized tires. And it's because there's not enough pressure pushing out on the hard rubber wall of the tire. Well, water does a similar thing in plants. As water flows into the cell, there's pressure to go into the cell because of diffusion. Substances tend to diffuse from areas of high concentration to low concentration. So there's generally more water outside the cell than inside the cell in terms of total concentration. So water's constantly wanting to come in, but it can't fill up totally and just burst the cell, because the cell is hemmed in by this cell wall made up of this semi-rigid cellulose. Because of this, then the plant has what we refer to as turgor pressure. And it's the pressure exerted by water on the cell wall that helps plants keep their form. How many of you have a plant in your office or in your dormitory? Anybody? Who has plants? No plants? Who's that?
Olivia.
Oh, Olivia, you have a plant? What kind of plant do you have?
I have an aloe vera plant and then a flapjack.
Oh, you have an aloe vera plant and a fly trap. So when you press on the leaves of the aloe vera plant, it's particular because it stores a lot of water in its stems and leaves, doesn't it?
Yeah.
Yeah. So if you denied any of your plants water for a period of time, what happens to your plant?
It would die.
Well, before it dies, though, what is it going to do?
It's gonna start to like kind of droop.
Yeah, it's going to do what we call wilting. Well, that wilting phase is basically due to you not providing enough water to your plant. As a result, the cells lose water, they lose turgor pressure, and as a result, they begin to lose their shape and form because they don't have this water pushing out against the cell wall to keep that form. That's the first sign that you should be watering your plants more, because the plant isn't dead yet. It's just not able to maintain its form because it doesn't have enough water to inflate its cells, push up against the cell wall, and maintain a particular shape. That's a plant-wide symptom of not enough turgor pressure in the plant cells. And turgor, I should probably write this on the board. One of the downfalls of teaching online is you forget to write things on the board occasionally. Turgor is spelled Turgor, and it's just this pressure exerted by water on the cell walls of plants. So then the last thing that you need to note that is different between plant cells and animal cells is plant cells have this special organelle called a chloroplast. And this chloroplast is green, it has an internal structure to it, and this is the site of photosynthesis. And we'll be talking a lot about chloroplasts and photosynthesis here shortly. Any questions about plant cells? All right. So when we look inside of a plant cell, this is a transmission electron micrograph. This is just a slice through a plant cell. You can see that there are, here's the big central vacuole, here's the nucleus. We have chloroplasts. Chloroplasts oftentimes have starch grains inside of them because one of the storage forms of glucose is starch. It's just another way of looking at the thing that you saw just a minute ago. There are connections between plant cells called plasmodesma that allows plants to transfer materials from cell to cell in spite of a cell wall being present. And so you can think of it as kind of like tunnels that go through the rigid cell wall that allow the cytoplasm of one cell to be in contact with the cytoplasm of another cell. And this is also how you can move materials from one plant cell to another plant cell. And this is crucially important in transport mechanisms. So when a plant generates glucose up in its leaves, it's going to store that glucose in the roots. Well, how does that glucose get from the leaves to the roots? Well, it goes through a series of cells that are connected to one another as water flows through the vascular tissues of the plant. So one way of thinking about plant bodies is we can think about the part of the plant that lives above the ground. This is oftentimes referred to as the shoot, and contrast that with the part of the plant body that lives below the ground, which is oftentimes referred to as the root. Plants grow from what we call meristematic tissues, and so we have plant meristems, and it's on the slide, so I won't spell it for you. But this is the portion of the plant that is undergoing active cell division. And there are a couple of meristems that you need to understand. At the tip of the plant, for dicots in particular, for this type of plant that has venation that kind of spreads out in the leaves, there's an apical meristem that occurs at the tip of the shoot, and there's an apical meristem that occurs at the tip of the root. There are also what we refer to as lateral meristems that occur where buds occur, and there is also a meristem in the vascular tissue. And so these are places where cell division is going on, and plants, just like you, grow through the process of duplicating its own cells. And so you go from a seed to a redwood tree through just tons and tons and tons of cell division. Leaves, which are these things, these are the primary organs of photosynthesis. And then these axillary buds are where you're going to get branches growing off to have the growth of lateral branches on a dicot. So this is a structure that is especially applicable to dicots. And we haven't talked about the difference between dicots and monocots, but we're about to. Questions about basic structure of the plant body with reference to the soil surface. So you guys hanging okay? All right. So another way of thinking about this is you can divide plants into tissues. Tissues, once again, have apical meristems and lateral meristems. These meristems then give rise to all of the different types of tissues that are found in plants. You do not need to know this figure. Let me repeat that. You do not need to know this figure. What you need to know mainly is that meristems produce all of the other tissues. The tissues that we are going to be talking about primarily are vascular tissues. And vascular tissues arise from both apical meristems and from lateral meristems, and they're made up of phloem and xylem. All the other stuff that's on here, if we were taking a biology class, we would expect you to know them, but we're not taking a biology class. I'm selecting the information that you need to understand certain things that I think are important for you to understand, and we're leaving a lot of plant biology behind in the process. So be very clear. Meristems produce lots of tissues. Tissues of interest to us are vascular tissues, xylem and phloem. Are we clear on that? Once again, I can't see you, so I need to hear you.
Yes.
Okay, at least one person is clear. All right. And I just told you that, and I lied to you. Skip this. I'm gonna delete this cell, this plant tissue slide, because I just told you I wasn't going to ask you about these things. So three slides we're going to delete out of this because I should not lie to you like I just did. So let's think about the vascular tissues. A xylem takes materials from the roots up to the leaves. And so you can think of xylem as going from root to shoot. It's made up of these different kinds of tissues that we just skipped over. And so it's referred to as a complex plant tissue because it's made up of lots of different types of tissues. But these are basically hollow tubes, essentially, that move fluids from the root up to the shoot. And in a similar way, phloem is a complex tissue, but it moves tissues the other direction. It goes from shoot to root. That should say root, not toot, for whatever reason. So it takes it from the leaves down to the roots. And once again, it's made up of sieve tube members, which are non-living fragments, remnants of cells and companion cells that are kind of the living cells that kind of govern the sieve tube members. So once again, a complex tissue made up of a couple of different types of cells, but the movement of fluid is from shoot down to root. When we look at these vascular tissues, they look different in monocots and dicots. So monocots and dicots are kind of two big divisions within plants. And in monocot and dicot, the cot refers to a cotyledon up on the board behind it. And these are sometimes referred to as seed leaves. When you plant a bean seed and it comes up, the first thing that happens is you have these two big leaves, and they're green, but they're really fleshy, and they don't have a lot of venation to them. And it's because they're not really true leaves like they're going to develop later. They're leaves that are part of the structure of the seed, actually. And in the case of monocots, there's one cotyledon, and in the case of dicots, there are two cotyledons. And so that's the derivation of the names. And one of the things that you'll notice in monocots and dicots is that the arrangement of the vascular tissues is different. In monocots, they're scattered all around the stem of the plant. And in dicots, they're arranged in a ring around the perimeter of the plant. And inside the plant is this centralized region known as pith. Here in monocots, the vascular bundles are distributed all throughout the pith, whereas here they're surrounding the pith in a ring of vascular tissues. So if you have a stem under a microscope, it's really easy to distinguish between a monocot and a dicot. Grasses are an example of monocots. Soybeans and daisies and things like this, sunflowers, are examples of dicots. So corn and soybeans is what you have here. And so one of the reasons we want to distinguish between monocots and dicots is because of this difference between corn and soybeans. When we look at their vascular tissues, we always find phloem to the outside of the plant. We always find xylem to the inside of the plant. So once again, we have phloem to the outside, we have xylem to the inside. You see the xylem has these big vessels that transport materials. And so in between the xylem and the phloem is the vascular cambium here. And so in plants that are dicots, you have the possibility of what we refer to as secondary growth, the plant getting bigger and bigger and bigger and bigger in terms of its girth. And that's driven by this meristematic tissue. So this is the lateral meristem that I was referring to earlier, is called the vascular cambium. So it's wedged between the phloem and the xylem, and it produces new layers of phloem to the outside, and it produces new layers of xylem to the inside. And so that's how dicots increase their girth over time, the thickness of the stem. Over time, you can get lots of growth in dicots, such that the lateral meristem is way out here, and it has produced all of these new layers of xylem to the inside, and all the new layers of phloem to the outside are all compressed right here at the outside perimeter of the stem, of the trunk of this woody plant species in this case. This is where the annual rings come from. The annual rings are made up of 1 year's growth of xylem. And so when you see the rings on a tree, it's because this vascular cambium has been producing layers of xylem over time. And then the phloem is squished in out here on the outside. Monocots and dicots also differ in terms of their root structure. So dicots tend to have what's known as a taproot, a central root that goes down that has a bunch of lateral roots that come off, whereas the monocots have what's referred to sometimes as either a fibrous or an adventitious root system. These roots all come from a common base, and rather than there being a strong central root, there are lots of roots that are all kind of the same size, and they spread out laterally as well as vertically throughout the soil, rather than a dicot root, which is primarily growing down, and then after it has grown down for a little ways, it starts to send out lateral roots. So once again, the way in which they harvest water and nutrients from the soil is going to be different because of the shape of the roots, and also the degree to which they're anchored firmly in the ground is going to be different depending on the root system. So once again, these are your grasses, and these are all the other main plant groups. All right, it's 10:02, and I wanted to just ask you here at the end, do you have any questions about the material that we've covered so far? Have you seen this in a biology class in high school at some point? Or can you remember back to high school at this point?
I can't remember.
Can't remember, okay. How many of you took high school in biology in high school? Okay, chances are you probably were exposed to something like this back then. So hopefully some things will start to shake loose in your memory. What we're going to do on Wednesday is we're going to talk about leaf structure, and then we're going to start getting into photosynthesis. So that's what's coming next. So like I said, the best thing that you can do for Wednesday is actually read chapter 3 of the Plants and Society book. All right. Then that's it for today, and I will see you on Wednesday. Let me know if you have any questions between now and then.